What if I told you that 80% of the genetic instructions causing disease have been invisible to medicine? For decades, we've been reading only 2% of your genetic code, the part that makes proteins. The other 98%? Dismissed as junk, evolutionary debris, genetic noise. When genetic tests found nothing wrong with your protein coding genes, doctors had no answers. But that junk isn't junk. It's a control system, regulating when, where, and how much protein gets made. And mutations there cause 80% of genetic diseases. Diabetes, autism, schizophrenia, cancer, heart disease. Now, scientists are racing to decode it, map it, read the instructions we've been blind to. And for the first time, they're finding answers, diagnoses, treatments, hope. Hello and welcome to Mind and Marvel. I'm Lucas, and I'll be your guide today. When scientists finished reading the human genome in 2003, they were shocked. Humans have only about 20,000 protein-coding genes. That's barely more than a roundworm. Roundworms have 20,000 neurons. We have 86 billion. How can similar gene counts produce such different complexity? The answer lies in the 98% that doesn't code for proteins. Early geneticists called it junk DNA. Evolutionary leftovers. Parasitic sequences that accumulated over millions of years. But hints emerged. Some junk regions were conserved across species. If it's truly useless, evolution should have deleted it. Mutations should have scrambled it, but it remained. Identical across mammals, even across vertebrates. The ENCODE project launched to decode this mystery. Encyclopedia of DNA elements. Test every region. Find its function. Map the dark genome. The results shocked the world. 80% of your genome is biochemically active, not making proteins, but controlling when proteins are made. How much? In which cells? Think of your genome as an orchestra. Protein coding genes are the musicians. The non-coding genome, it's the conductor, the sheet music, the acoustics, the scheduling. Musicians without a conductor play chaos. Same notes, wrong timing, wrong volume. That's disease. Right proteins, wrong regulation, wrong expression patterns. The non-coding genome contains regulatory elements, enhancers, silencers, promoters. They bind to genes, turn them on, turn them off, fine-tune their activity. It produces long non-coding RNAs, thousands of them, regulating gene expression, microRNAs, tiny molecules, each controlling hundreds of genes simultaneously. It organizes chromatin structure, the 3D architecture of your genome. Genes physically move closer to or farther from their regulators. Space matters. This is the hidden control layer. The dark genome. And mutations here cause most disease. Meet Sarah. Severe developmental delays. Seizures. Testing every known gene. All normal. Meet Jake. Type 2 diabetes at 25. Family history positive. No mutations in insulin genes. Meet Emma. Autism spectrum disorder. Standard genetic panel? Negative. A pattern emerges. Obvious genetic diseases. But protein-coding genes are normal. The dark genome holds the answers. Example 1. Mutations in enhancers controlling limb development genes. The gene itself is perfect. Makes normal protein. But the enhancer is broken. The gene doesn't turn on during limb development. Result? Limb malformations. The gene is fine. The regulation is broken. Standard tests miss this completely. Example 2. Type 2 diabetes. 90% of genetic risk is in non-coding regions. Enhancers controlling when pancreatic cells make insulin. How much? In response to what signals? Mutations disrupt this fine-tuned control. Insulin production becomes dysregulated. Blood sugar control fails years before diagnosis. Starting from regulatory mutations. Example 3. Cancer. The most famous example. Oncogenes exist in everyone. Genes that promote cell division, normally tightly controlled. Mutations in regulatory regions. Silencers break. Oncogenes turn on inappropriately. Cell division runs wild. Tumors form. All from regulatory mutations. Genome-wide association studies reveal the scope. 80% of disease-associated genetic variants are in non-coding regions. GWAS, Genome-wide association studies light up the genome. Most signals? Regulatory deserts, far from any gene. We've been looking in the wrong place, reading the wrong 2%. Neurological disorders, autism, schizophrenia. Most risk variants are regulatory. Brain development requires exquisite gene timing, right cells, right moment, right amount. Regulatory mutations throw off timing. Neurons form wrong connections. Neurodevelopmental disorders emerge. Autoimmune diseases. Immune system genes are tightly regulated. Regulatory mutations cause inappropriate immune activation. The body attacks itself. The common thread? Regulation gone wrong. Not broken proteins. Broken control systems. Reading the dark genome requires new technology, new approaches. First, whole genome sequencing. Read everything, all 3 billion letters, not just the 2% coding for proteins. Cost has plummeted from $3 billion in 2003 to under $200 today. But reading is only the first step. Understanding requires interpretation. Second, machine learning. AI trained on millions of genomes, patterns emerge. A regulatory grammar, rules for how enhancers work, which sequences matter. AI can now predict regulatory function from sequence alone. Which regions control which genes? An AlphaFold moment for the regulatory code, cracking the language. Third, functional genomics. Don't just read, test. CRISPR allows editing specific regulatory regions in cells. See what changes. Massively parallel reporter assays. Test thousands of regulatory sequences simultaneously. Which sequences boost gene expression? Which silence it? By how much? Building a functional map. Not just sequence, function. Fourth, single-cell genomics. Different cells use different regulatory programs. A liver cell and a neuron have the same genome, but completely different active regulatory regions. Mapping which regulatory elements are active in which cell types, which diseases affect which tissues, tissue-specific disease mechanisms emerge, why diabetes affects pancreas, why schizophrenia affects brain. Fifth, clinical implementation, bringing dark genome analysis to patients. Diagnostic labs are adding regulatory region analysis to standard testing. Remember Sarah? Developmental delays, normal genes, whole genome sequencing found it. Mutation in an enhancer controlling a critical brain development gene. The gene was perfect. The on switch was broken. Diagnosis after years of mystery. Diagnosis changes treatment. Regulatory disorders need different approaches than protein disorders. Some are amenable to small molecules, drugs that restore regulatory balance. Others to RNA therapies or targeted gene editing, fixing the regulatory code itself. Understanding the dark genome enables entirely new therapies, antisense oligonucleotides, synthetic RNA sequences that modulate gene regulation, already FDA approved for some diseases. Nusinersen for spinal muscular atrophy. It works by altering RNA splicing. A regulatory mechanism, not fixing the protein, now expanding to direct regulatory enhancer targets. Small molecules that activate or silence specific enhancers, turn up beneficial genes, turn down harmful ones, restore regulatory balance. CRISPR-based regulatory editing, not cutting genes, editing enhancers, epigenetic editing, changing how DNA is packaged, making regulatory regions accessible or inaccessible. CRISPR and CRISPRi, activation and interference systems, turning genes on or off without cutting DNA, potentially safer than traditional gene editing, reversible, tunable. Clinical trials are beginning, regulatory therapies for previously untreatable conditions. Autism spectrum disorder, restoring proper timing of brain development genes. Diabetes, fixing pancreatic insulin regulation at the source. Cancer, silencing oncogene enhancers, preventing inappropriate activation. The future is personalized regulatory medicine. Your regulatory mutations mapped. Therapies designed specifically for your regulatory disruptions. Not one size fits all. Precision regulation. Tailored to your dark genome. For a century, medicine focused on proteins, the 2%, the visible genome. But proteins are just the instruments. The dark genome is the conductor. 80% of disease was hiding in regulatory shadows, invisible to standard testing, now emerging into light, mapped, understood, targetable. Patients who spent years without answers are getting diagnoses. Some are getting treatments. The revolution is beginning. But we're still in the early stages. Most of the dark genome remains mysterious. Millions of regulatory elements, most uncharacterized. Each person carries unique regulatory variants. Your dark genome is unlike anyone else's. Understanding it is understanding what makes you susceptible to disease and what protects you. Which leaves us with a profound question. We've called DNA the blueprint of life, but blueprints are static, fixed. The regulatory genome is dynamic, responsive to environment, to experience, to choice. Your experiences literally change your regulatory landscape. Stress, diet, exercise, trauma, all alter gene regulation. So are we determined by our DNA? Or do we continuously rewrite our regulatory code through how we live? Is the dark genome the interface between genetic destiny and free will? The place where nature meets nurture? And if we can deliberately edit it, rewrite our own regulatory code, what does that mean for human identity itself? If this revelation about the hidden 80% of your genome fascinated you, hit that like button. This is medicine's next frontier. Subscribe to Mind and Marvel for more deep dives into the cutting-edge science rewriting what it means to be human. Drop a comment. Would you want your entire genome sequenced, including the dark 98%? What disease mystery do you think the dark genome will solve next? Share your thoughts. This is Mind and Marvel, where science, survival, and the superhuman collide. Until next time, remember that you're more than your genes. You're the regulatory code controlling them, and we're just learning to read it.